I don't think I really need to introduce Chris Evelyn to anybody here, other than to say he's had an illustrious career, which we can slightly review. He got his undergraduate degree at Loyola Marymount in classics and urban planning. He discovered that speaking ancient Greek was not a marketable skill, so he came to Bren and discovered that didn't provide marketable skills either, so he switched over to the EMD and has been working in my lab for a couple of years now at least. I forget how long. I think everybody here recognizes that Chris has been one of the most active and involved grad students we've had in terms of departmental affairs and in terms of helping other people out. I don't think there's a person in the room he hasn't helped in some way, often extensively and often in ways that he didn't really have to, but he did. So we would miss him, except that we're not going to really have the chance to miss him, because he is going to be around for a while longer, and I'll explain that in a moment. By way of introduction, I would say that Chris is going to be talking today about what I would call the crest of a third wave of analysis of a really interesting system that has occupied the careers of a whole bunch of people in California for the last 40 years or so. And that is that starting in the early 1970s, Dave Wake's lab at Berkeley began to look at the radiation of slender salamanders in California, which at that time was regarded as consisting of a single species in California and one in Oregon. A single species in California was regarded as being highly variable, and we thought that was probably not true. But after a number of years of extensive field work and using at the time electrophoresis, which was the state-of-the-art technique in the early 70s, Kay Yonov and others ended up discovering that there are about a dozen cryptic species in this group. And that not only that, but the distribution of these species corresponded in strong ways to a lot of the tectonic history of California. In other words, these species are old enough that they had actually moved around as pieces of landscape and shifted: San Andreas Fault, Salinan Block, and so on. And that went as far as it could with the evidence that we had until about the 1990s, when Elizabeth Jokish and her colleagues started doing sequencing work. And since that time, the sequencing work which has been done has resolved what we think is probably a pretty accurate phylogeny, so that we have a good idea of how the species are related, and also a pretty good idea of how many species there are. And there are 22 now, as opposed to one before, and a number of others that are suspected or known and not yet fully documented. So what is happening now is, with that stable phylogeny and with that understanding of the evolutionary history and the geologic history connecting to it, we are finally able to address a bunch of questions about the ecology and phylogeography of these salamanders. You can't do it if you don't have a good phylogeny. And that's what Chris is on the crest of the first wave of sort of post-descriptive analysis is doing. And the results of that are really getting to be quite interesting, so we're sort of looking forward to it. I should point out that joining us by Skype is the third member of Chris's committee. Steve was the second. Elizabeth Jokish, who is the architect of much of what we currently know about Batrachoseps, so welcome.
I'm on mute, so we don't have to.
Yeah, she can't hear me.
She can hear you, but we can't hear her.
Oh, that's too bad. Anyway, we will have a little reception following us upstairs. And I just wanted to comment that in the last few weeks, Chris and I managed to pull down a grant that's going to keep him around for a while as a postdoc. And that it's likely that he will also be teaching vertebrate biology in the fall for END. So, very happy to have him finally finished. And I'm looking forward to what he has to say. Chris.
Thanks, Sam, for the great introduction. Hopefully this works this time. Yes. Oh, except this is like half of the talk. Okay. Hi, everybody. Thanks for being here. It's always a real pleasure to speak here and get eyes on stuff I've been working on from so many smart people and get their critical feedback. And also a pleasure to talk to friends and family over more than just beers and food, which is how we usually talk about this. and briefly. As Sam mentioned, we're going to talk about slender salamanders today, and obviously I went for the hard sell here, including the Darwin's finches on this first slide, and we'll get into that a little more as we go along. Before I go any further, I have a lot of acknowledgments. There's probably more people I could put on here. First of all, my committee and my family. I couldn't have done any of this without my family, and they're here. Darwin and Lori for living with me through this, and my parents and my sister, who's not here, for the long trips through the reptile house at the LA Zoo as a kid. I'm sure it was very tedious for them, but really enjoyable for me. And obviously my committee, Sam, gave me an amazing opportunity here. I was not probably the most traditional student to come in, and he gave me a chance. Elizabeth has been a wonderful collaborator from afar, hosting me in her lab and letting me join in her field trips when she was out here. And then Steve Rothstein welcomed me into his lab. I had a lot of my ideas for the first time. I presented to his lab and his meetings to his students and got some really great feedback. So all of you have been just really great. Thank you for that. There's a ton of other people on here. I had field help from a whole bunch of people, including some people who are here. I think Brent's here, a lot of other names I recognize. The MB grad students, and I have to say, you guys are awesome. Even today, it's like last minute I'm scrambling, and I just grabbed Louise into like helping me do this and that. And but just overall, so many of these ideas I started, and then I didn't know how to actually execute them, and you all stepped up and told me how to do it. I see Mark especially, and Sabrina Pankey, both of them in particular helped me. As well as I talked to the Oakley lab, the evolution group. I mean, I could go on and on. This would be really fun to just thank people all day. I really could do it. But, and then all the landowners and managers who aren't here, but the Katanis in particular, who I kind of got started doing this on their land. The Tohono Ranch Conservancy, the Nature Conservancy, and Tom Maloney and Mike White at the Tohono Ranch Conservancy especially were supportive. And Nature Conservancy also with some funds and access. A ton of advice. Also, Sam mentioned Dave Wake. It's actually Dave's 80th birthday today. So anyone who knows him, you can go ahead and send him an email and say hi. He'd probably love to hear from you. Dave and Marvelly, both of them, have been really supportive when I got up to talk to them. And that's just been really, really great to build that relationship with the folks up at MBZ. And then Steve Davon, who's another MBZ affiliate and head patent and Frank Davis here on campus, who's my master's advisor at Brent, and has continued to give sage-like advice. And then wider friends and family, including Robin Williams at the bottom. That's not Robin Williams who just passed away. That's actually my 6th grade science teacher, who's like the one teacher I ever had who liked having me in her class. So. Anyway, and then there's a picture of the animals. We have to acknowledge them. Many animals were harassed and sacrificed over the last 10 years at my hands, and hopefully they live on in immortality and that their deaths were worthwhile. So I'm going to just start, biodiversity. This is one of these great shots from this National Geographic photo art project that they have going on. And the diversity of life, I think most of the people in this room is something we just find inspiring and just awe-inspiring and really impressive. And these are great photos if you get a chance. It's Joel Sartori, I think, is who does all the photos. Obviously, I'm disappointed. There's only 2 salamanders in here out of all of this. So I wanted to talk more about salamanders today. The largest family, most specious family of salamanders is the plethodontidae. There's over 400 species at this point, and there's probably still going to find some more. There's 3 main centers of diversity in Central America and Mexico, Central America, South America. There are around 300 species, and here's some. They have all kinds of weird little morphologies: webbed feet, giant nostrils, all sorts of cool stuff. And these are all fully terrestrial. So the plethodontidae are marked by being lungless, first of all, so they do not have lungs. And secondly, for having a lot of species that are fully terrestrial with no aquatic life stage. In Western and Eastern North America, we have about around 100 species. And there we actually do have species that have aquatic larval stages. And you can see these are all semi-aquatic species that have these compressed tails. And so they are semi-aquatic and larval fully aquatic. And then here in Western North America, we have about 40 species. And all of the species in the West are fully terrestrial. So they have no aquatic larval stage, and they live entirely on land. And you can see around 40 species, so as Sam mentioned, about over half of those are Batrachoseps. So as far as I can tell, they're the most speciose vertebrate west of the Mississippi. Haven't found anything else. And one of the other cool things about studying plethodontid salamanders and Batrachoseps is we're still finding species. And these are two of my very favorite books to just leaf through, just open to a page, and there's always just beautiful nuggets. John Steinbeck, Log from the Sea of Cortez, and The Voyage of the Beagle. And there's just something innately exciting about discovering new species and new populations. And so I highly recommend doing it at some point in your life. Going and looking somewhere where you think there might be something new and finding it's very exciting. And so the type of diversity that I'm going to be talking about today is between species in a related group. So interspecific variation. And you can also think of this as variation on a theme. So we have some classic cases that are familiar. Darwin's finches, right? There's none of these finches that grew antlers or breathed fire or swam in the ocean, but we see differences like beak size, differences in overall size that you start to ask your question, what led to this pattern we're seeing. There's lizards, the animals, which have got a lot of received a lot of research interest lately. Differences in color, limb morphology. Here in California, we have oaks, again, variation on a theme. They all have, leaves, a lot often spiky, these acorns, but there's slight differences. And then stickleback, which respond to abiotic and biotic conditions, and to evolve different morphologies. So here in the Batrachoseps, we see, again, variation on a theme. These are, range from very worm-like species with really small limbs and feet to more generalized forms that look like a respectable small terrestrial salamander. So just a quick overview, and Sam touched on some of this. There's around 22 species. We think there's probably a couple more. In California, Baja, and Oregon, they're all fully terrestrial. They eat small invertebrates, and they're seasonally abundant. I'll come back to this phylogeny a few times during the talk, but at this point, I just want you to see two things, just kind of see the number of species, and then that there's two subgenera, and that these three species in this subgenera, you have this deep division here. This Plethodontid are the species in black, so here's 1, 2, 3 species. You see this disjunct distribution, and then the subgenus Batrachoseps, which is the other 20 or so, 19 or so species, are in these yellow dots, which cover coastal and inland foothills of California. As I said, they're fully terrestrial, so you find their eggs, if you're lucky enough, which is rare in a lot of places. They'll be in a wet little spot of soil, and here's when the eggs hatch. You get this little miniature salamander coming out. Seasonally abundant, so here's a hillside where actually you find Batrachoseps. It's not the typical, maybe, eastern North America habitat. You think of like a wet forest. Here we have juniper, we have some yucca, this kind of rocky hillside, and if you come there most of the year, you won't find anything. If you come there at the right time of year, every one of these rocks has salamanders under it. So kind of an interesting pattern as we watch them respond to surface moisture. They feed on small invertebrates, and I'll show you this one. I have one cool little video here. So this is really, really slowed down. And they fire their modified epibranchial bones and grab little invertebrates with their tongue. So that's how they all feed. So again, they have all these things in common. So why are Batrachoseps interesting to work on for questions of interspecific variation? Well, one has been interesting reason to work on is because, like I said, we're still discovering species. Here's this great phylogeny from the late 60s, based largely on morphology. And then here's the one we have today. Homoplazy can be helpful if we're thinking about things in evolutionary terms. So if we have species like spread across the phylogeny that look very similar, but there's really no phylogenetic signal in their morphology, then we can start to ask questions about, is there something about their ecology that's similar, of these species that look similar? Last, dancing horses, and I've told this story before, but on the Catani's ranch 10 or so years ago, I was invited to a party that was about the best party I've ever been to. We were laying under the blue oaks, drinking beer, listening to a mariachi band while somebody has a dancing horse and rides around on a dancing horse. And I asked Sam what I should do when I go up to Kern County to this party, and he said you should look for this sort of rare Batrachoseps. And when I first saw it, I had seen it in a book, but for some reason when I first had it in my hand, it was so different from the things we had here. It really got me wondering about these questions of, why would this look so much different than what we have here in Santa Barbara? So I'm talking about three sections from my dissertation. The first, what is the ecology of Batrachoseps when their ranges overlap? Second, does morphology predict range size? And talk about the implications of that. And last, has rollback morphology involved in response to seasonal climate that we have here in most of California? So first, what happens when species of Batrachoseps, when their ranges overlap? And here's the pair we're going to talk about. This is that more robust species I talked about with the dancing horse, and its range is this small area here from about the San Andreas Fault Trace up to the start of the Paiute Mountains, just south of the Sierra Nevada. And this other species here, the more slender species with smaller limbs and longer tail and longer body, is Batrachoseps nigriventris, and that's what we have here in Santa Barbara from up San Benito, Monterey County, all the way down through the LA Basin. And so here we have this comparison between a worm-like species with a large distribution and a more generalized species that's restricted. Overall patterns in their biogeography, unlike we see in the Northwest, where we have 2 to 4 species of plethodon in the same genus, overlapping and found together in the same area. Here, Batrachoseps really replace each other, as you go along. So one species replaces the next. There's very little overlap, and this, this suggests ecological replacement, and that's sort of how this has been interpreted. But it hasn't been really formally looked at yet. So that's what I'm going to talk about a little bit in this section. There's also some challenges to doing this work, especially in the area right here where I was working. It's very remote, it's hard to get around, and in some other cases where Batrachoseps overlap, the species look really similar, and you cannot identify them, even if you have them in hand. Even an expert can't tell them apart. So there are some challenges to doing this, which is probably why it hasn't been tackled before. So what sort of expectations do we have about when these species overlap? First, the ranges might overlap, but they actually might not be sympatric. So there might be this broad partitioning of this habitat where the ranges overlap. If they are found in sympatry, we might see spatial partitioning, temporal partitioning, or that allow them to, because they're using a lot of the same resources in terms of cover objects, in terms of food, that you avoid competition through these. And then, or there might be behavioral interactions to mediate this use of resources and agonistic behavior. So this comparison, as I said, they have a close phylogenetic relationship. So you kind of controlled for any phylogenetic influence on their behavior and their ecology. So historically, in this area, we have a disjunct range for Batrachoseps stevensi, which is the more robust species. We have specimens from the south near Fort Tejon and on Tejon Ranch, and then a few specimens up on Caliente Creek north of Tehachapi. They thought Batrachoseps nigriventris is kind of broadly found from 400 meters to 1,100 meters in elevation. And Batrachoseps stevensi in this narrow band. So again, we have this narrowly distributed species, and it looks like, wow, it really is a specialist. It's really only found in this very narrow elevation band as well. And they have been found at Syntopi in a couple locations, but rarely. So anyway, the goals for this field study were do they occupy the same microhabitat in the field, and then I also did a test for agonistic behavior in the lab. So I had conducted field surveys for 4 years and looked at microhabitat temperature, moisture, cover object use, and then elevation of their localities. It's a test for agonistic behavior. I did arena trials in the lab. So a lot of these agonistic behavior trials have been done on eastern plethodons, and they're actually pretty exciting. So they have ratings from overt aggression from 0 to 6. 6 would be something like biting or chasing, and then and for aggression, and overt submission would be ducking and pressing their bodies against the substrate, trying to look small and non-threatening. So there's a good basis for agonistic behavior in this family. And anyway, doing field data, the fun stuff, hiking up steep hills, pulling open logs, looking for critters, stuff we all love to do. Anyway, so first of all, I did get quite a bit more data, although I did not find as many as I would have liked, which is probably always the case for everyone. And the distribution of Batrachoseps stevens is still disjunct, although there was quite a bit of survey effort in this area, we just didn't turn any up. But the results of this, so the overlap between these two species occurs at a pretty narrow band of elevation from 1,050 meters to 1,200 meters. But Batrachoseps stevens is not so restricted as we thought. It actually extends its elevational range up to 1,800 meters. Batrachoseps nigriventris was found, the worm-like species was found in drier and warmer habitat, but the overlap was considerable. And temporal partitioning, which was probably the coolest result, and I wish I had more of it, was seen at one site where I went in March when it was wet and cool, found Batrachoseps stevens, the general species, went 2 months later when it was drier and warmer, and it was the worm-like species, Batrachoseps nigriventris. And just to give you an idea of how the data played out, here's the elevational split. You can see that's significant. And even though some of these other were statistically significant, there's a whole lot of overlap. So really not much evidence this is a big difference in these factors. The arena trials, anyone's ever found a Batrachoseps, you can imagine, they were not very exciting. And actually, to review the data, what I ended up doing is taking videos of everything. I had to fast forward it 4 times just to even notice when they moved, because they just move so slow. You just can't even tell. They're just, if anything's happening. But this picture here just kind of sums it up. This was really common. This is the larger Batrachoseps stevens and the smaller nigriventris, and they're just sharing a cover object that's in there. So this was common. They didn't have any, they didn't really have any mind of each other at all. So yeah, nothing exciting. It was like until 3 in the morning just watching them not do anything. So conclusions of this. So the microhabitat use really overlaps considerably. Habitat is broadly partitioned at elevation. Aggressive interactions are unlikely to play any role in what's going on. But as I said, with that temporal partitioning, what would be really cool to do now is go back to these sites where we know they occur either precisely together or really close to each other, and see and sample across the season and see if their timing is a little different. You get a better idea of that. Okay, so once we had that, I said, okay, well, so the ecology and the microhabitat use does look pretty similar, even when we look at these morphologically different species. So the other thing we have this pattern of elevation use, and there were a couple other instances where I looked at the data. I said, oh, this is neat. We have a worm-like species at low elevation, and as you go up in elevation, you get more robust, more generalized forms. So that's sort of suggestive. And so, so I looked at range size and also elevation with these species. So species range size is interesting for a couple reasons. It's a fundamental unit of biology. It has been for a long time. Just the way we think about species. One of the first questions is where are they found, and it's generally has a log normal distribution within a clade. So if you look at a group, you have very few species that have a really large range, and then many more that have medium and small ranges. It's significant for macroevolution and for conservation, because both of those fields are really interested in extinction and why it happens and how often it happens and what are some traits, if there are any, that lead to one species being favored for extinction. So I took a look at this kind of in this context, and here's a diagram just about how it relates to macroevolution. So if you look at lineages through time and you think about how diverse they get, and how many species, which is what this is measuring. So we have these different groups of fish here, and these patterns have fascinated evolutionary biologists. And why does this happen? Are there traits that lead to this? Is this random? Are these random events that happen that lead to this pattern? And you can think about this the same way with populations in a clade or a number of just individuals in a species. And in the context of the North American West, and amphibians especially, this is really is really a straight line comparison, because what we see in the West is we've had this interglacial era with drying, and we've had orogeny of these mountains and a Great Basin spreading, which has led to drying inland conditions. And we see these species that have been isolated. And so here, this is one genus, Aneates, which is mostly distributed along the coast, but there's one species out of New Mexico just at the top of a couple mountains that's just stuck to these sky island species. So if you look at range size, you can see how that directly relates to, wow, that species is probably on its way out, right, if these conditions. And plethodon, you have the same. We have a coastal northwestern species, but then up in these wet areas of Idaho, we have one species here and then another species in on a sky island in New Mexico. And so this is, directly relates to how we think about, how range size is important in terms of extinction for both conservation and macroevolution. And the nice thing about Batrachoseps is because we have 20 species, we can look at this and be a little more robust with our statistical tests and easier to draw conclusions. So the first question, is evolution of morphology correlated with evolution of range size? And the first thing we need is a really nice metric, and this was Inigo Martinez Solano, who was at MBZ and measured probably 1,500 Batrachoseps. Batrachoseps are mostly smaller than my pinky, so this is pretty tedious work. And you take 9 measurements on each individual. I came along a few years later, I finished the last couple species that he hadn't finished, and so we can look at the whole phylogeny. What we did for this is take all those linear measurements of the body, kind of standard head width, body length measurements, and then put them in a principal components analysis, use the principal component 1, and then regressed it against snout bent length. So you're controlling for the size of the animal, and you're talking about differences in shape. So a difference in the shape of one. And you can see the way it came out actually looks like it's pretty effective, right? So we have these generalized species down on this end, and then as you go to lower numbers, we have more worm-like species. So looks like a metric that's going to be useful for us. For the range size metric, I played with this a little bit. So this is sort of the standard map that you see, maybe some points of where animals have been found, a hand-drawn polygon by somebody. I thought that was pretty good. And so I used both those metrics to, for latitude extent, you just take the lowest latitude and the highest latitude. That's one measure of range size. You have the polygon area you can measure, but then you can also do these, use these triangular irregular networks to draw triangles between all the nearest neighbors, and then you have a cutoff for how long of a line that you'll allow to stay to keep that population. And this, actually the colors didn't come out great for this, but basically it does a little better job picking up disjunctions in the range. So here's a gap where there's a big dry valley where there are no salamanders, and here's another gap here. And it's just a little more explicit with just talking about how you're measuring the range. So I used all 3 of these just to kind of get a robust conclusion. And then here's the phylogenetic hypothesis I mentioned before. This is from Elizabeth's paper in late 2014, using 5 nuclear markers. Okay, so the results. All the range size metrics were highly correlated, so that was good. That's what we expect. And the evolution of worm-like morphology is correlated with evolution of increased range size, and pretty significantly so, with a piercing correlation of -0.75. So more worm-like species have evolved larger ranges, and that's independent of phylogeny. So that's cool. So, but how are they doing this? Like, did they evolve some other trait that we can measure or some other ecological pattern that can help explain this? And the 2 that I thought of, one is, did they evolve to occur across a large elevation range? So you can see how being across a large elevational range would help you have a larger species range size. And then secondly, or are they occupying more abundant ranges? So are they occupying lowland areas, which we have more of, versus mountainous areas, which we have less of? And the first one is analogous to this niche breadth hypothesis. So just that if you have a wider niche, then it's easier for you to occur in more places, kind of think of something as a generalist. The second with the habitat availability hypothesis, so how much is available that could also drive you having a larger range. And the answer was no in both cases. So the evolution of these things was not tied together. And so that was kind of interesting also because it didn't follow through these patterns. So in the end, range I think correlated with evolution of world morphology, but not with occupation of a broader elevational range or of more available, more abundant elevations. So then this is the question, right? So for the macroevolutionary question, even for the conservation question, how do these skinny worm-like winters happen, right? How do we get skinny winters? We have these species that occupy tens of thousands of square kilometers versus species that's found on just tens of square kilometers in some cases. So it's really a large difference. And is there a shift in life history that's happening between worm-like species and more generalized species? So now I'm going to talk about climate seasonality and morphological evolution. So as I started looking at this, a couple models came up. First of all is this, I just call it the fossorial lizard model. So this is this Carl Gahn's idea from the 70s that you evolve to be more elongate. So this is the big question, like going from lizards to snakes or from limbed lizards to limbless lizards. Because you become more fossorial. So there's selection to have a small diameter, there's selection to get rid of these limbs which you're not using and use more undulatory locomotion. And in small lizards like skinks, this is a pattern that we see repeatedly across that family. And here's a sand swimming skink from Florida. And you look at the Batrachoseps and you go, oh, maybe this is something, this is something interesting. And I took some Batrachoseps down to Florida and I videotaped them walking and tried to see if there was a difference between the robust ones and the skinny ones. And they actually, I don't have the video here, they actually use their tail a little bit to press against the ground like a, fifth, sixth, seventh limb. They'll have these multiple contact points. They actually kind of push themselves along, it looks like, with their tails. And I said, oh, well, maybe this really might be something, that we have these things that are, look like they might follow the same model. Now, Batrachoseps don't actively dig, so they'd be just sort of swimming through leaf litter, but still, I had some hope that this would be. The other hypothesis that came up is the idea that elongation in some small vertebrates is due to selection of fecundity. So that if you have a longer body, you can hold more eggs, and because you don't have the girth anymore, that you need to have a longer body just to carry eggs, especially on the female, to hold eggs. And again, some skinks that are here in North America, the plethodontid skinks, have been looked at in this pattern by Griffith, and Elizabeth Jacques actually found some evidence for fecundity selection in Batrachoseps. So here's another hypothesis. So my hope is to figure out a way to pit these two against each other in some sort of a test. Again, fully terrestrial lifestyle we talked about before, so these things are kind of like lizards, and they occupy lizard habitat. And just thinking more about what a Batrachoseps is doing over the course of a year, there's the seasonal moisture. You can find them near the surface, climbing around, eating bugs, probably trying to mate, although we haven't seen that in a while. And as you get in the substrate, just thinking about this is kind of a nice figure, you have all these little facets, little rocks, root channels, places where they can hide and get down into that remain relatively stable in terms of the temperature and moisture. Here's a figure that demonstrates this, but this is in a less complex habitat. This is just in a sandy desert. And you can see this general thermocline for temperature, and then for moisture across the year. So this goes from January here to December. And so if you can remain below this dashed line in terms of depth, as an amphibian, you're not going to desiccate. But if you get stuck above it, then you are. And so just this idea that over the course of a year, a seasonally active amphibian is having to find this depth and then come back up when food is available and when other animals are available for mating. So this is basically what we think a Batrachoseps is doing over the course of the year. Very difficult to observe. If you dig in a spot in the summer, you will not find Batrachoseps. They're gone. You will not find them. It's been tried, but this is what we think is happening. Okay, so this question was tackled. At least kind of a framework for it was created in the 70s by Virginia Majorana, who's his wife of the, who she married? She was married to Lee Van Valen. That's right, she was married to Lee Van Valen. And so she looked at this, she said, okay, so we have to have an arid climate is selection to be fossorial, right? So there's selection against surface activity, there's selection that you need to be skinny and be stable of the surface. If food is available only periodically, then you're going to have selection for fecundity because you're going to have, probably a high mortality rate for your young. And if you have less predictable surface activity, you're going to need to store lipids, and that's what this big fat tail might also come in handy. So, is this morphology, this worm-like morphology, due to them being fossorial, or are they just trying to be seasonally active and maximize their fecundity and their lipid storage? And the framework she looked at this was actually really interesting. This was right about the time that Stephen Stearns and others were, let's say, dismantling, I guess, this really hardcore R versus K selection paradigm and talking about optimization. And so what Virginia did is look at how seasonal are these climates. When will these species actually be able to be at the surface? And she found, really to no surprise to many of us here, the activity is really seasonal in California everywhere. You have really dry summers generally, and you have wet winters that are not all that predictable. And so she came up with this framework, but couldn't really go too far beyond being descriptive of this. But one other thing that she did, which was cool, was she quantified how predictable these climates were and how seasonal they were. And she used this statistic, this Caldwell's index. And I'm just going to do a quick explanation of it here. But what it attempts to do is give you an idea of how predictable a climate is. And so there's two axes. So we have a time axis. So think of this as, this is a tree species A. And so let's say we sampled across 3 seasons, and we're trying to capture the phenology of the species. When do they flower? When do they fruit? And so for this example, for this species A, as we move along the time step here from one season to the next, that has no information for us, right? And that because it's always FF, which is flowering and fruiting, FL flowering, not flowering or fruiting. So if you go across the time steps, it doesn't tell you anything. So the time, your M, which is our seasonality, doesn't tell us anything. But if you know that you're flowering, you always know how much data is in that node, if that makes sense. Here's the inverse, right? So we have flowering and fruiting, non-flowering and flowering. Being on a time step tells us everything, because if we know what time step we're in, we know where all of our data is. We know all of our data is non-flowering. It's perfect predictability. You can also get to perfect predictability by having all of the data known by a sum of these two. By a sum of seasonality, and this is called constancy. So that is kind of the framework, but the things we're going to look more at is that our predictability is going to be less than 1. It's not going to be perfectly predictable, because if we're in season 1, let's say we have most of the trees are not yet flowering, but maybe one is. As you move on through the seasons, more species are flowering and fruiting, but it's never perfect. So even if you know the state you're in and the season you're in, you don't know exactly, it's not perfectly predictable. And so really what this just gives us is a way to quantify how seasonal and predictable the habitat is, and in this case we're going to use rainfall. So you pick bins of rainfall. It goes 0 to 32, it increases. And so we see if we have, so this is our months of the year. What does she have this starting at? September. So she has starting in September, and you can see if we know we're in September, we have our mode is that, we get 1 centimeter of rain. But there's some spread here. And as you go through the year, that spread moves along. And then you can quantify for the full year how predictable the climate is, how constant it is, and how seasonal it is. And one thing while we're here, I just want to point out is, one thing is, because you're using bins here, you can you can get a result where it's just as seasonal by going from really low rain to a little bit of rain as you can from going from a lot of rain to a whole lot of rain. So you have to keep in mind where you are along this. This doesn't tell us everything, it only tells us how seasonal it is. So if you want to, and I'll explain more why I care about that. Okay, so in looking at this hypothesis, I thought she had a really nice framework for looking at it. But as we saw with her her map there, she really only had it's kind of 3 data points to talk about this. So now we have all these big advantages, right? So we have spatially explicit climate data that we can use to test this. We have a resolved phylogeny, which Sam was mentioning, which we've talked about before, so we know species relationships. We have phylogenetic independent contrast methods so that we can really control for our phylogenetic influence. Better locality data. We have our statistic to quantify the morphology. And we have E and B grad students. Like, so Mark Wilbur helped me so much with this. I came in with an idea and I said, Mark, how do I do this? And he said, no problem. And he took him like 20 minutes and he gave me this code. It was great. And of course, we also have the indomitable Tom Smith here. This is all since 1976. These are all the improvements that we have had. So, compare, here's from her original paper where she had some samples. And so, but the data that I was able to use here is basically have those climate layers, and it was 1981 precipitation records on a 4-kilometer grid from 1981 to 2013, I think I used. And you can just drill down across those layers for each of these localities, for each species. So you're getting basically a bunch of data for each species that then you can use to make comparisons. And here you can see kind of obviously the blue, purple are the wetter areas, and the yellow, red are the drier areas. And there's a ton of ways you could have chopped this up. And what I chose to do is think about the context. If it's more arid, they're going to be more fossorial. That's where the selection is going to be. If the food is available periodically, it's highly seasonal, then that's going to be more selection about life history and the lipid storage we talked about. And then I also looked at predictability. One thing that cannot be overlooked is that if we're going to talk about when species are active, we have to have some idea about their ecology. We have to have some idea about their behavior. And a ton of field work has been done by Virginia Majorana in the 70s and Hendrickson before that, just to figure out that there's sort of this below 6 millimeters of rain, there's no activity. So in this study, I defined this as the dry season is less than 6 millimeters of rain. And so that is really critical, and all that good natural history field work that was done is really critical to being able to pull something like this off. We also expect selection to happen at the extremes. So, in that northwest California where it's very wet, this is probably not a big selection from, as it's not probably as big a deal to have these seasonal patterns because they're going from a lot of rain to a little bit less rain, but they're not going from no rain to a lot of rain. So I started with the most xeric 25% of that data, and I'll just give you a snap. This isn't too much of an eye test, but basically the shaded in gray are the dry seasons, and I just took consecutive months that were under 6 millimeters of rain. And you can see there's quite a bit of variability in the length of those dry seasons, but also that by taking the driest 25% of the habitats, we accomplished the goal of we have dry seasons where they're inactive and the wet seasons where they're active. So first, here's some results. So here are the metrics for the dry season. How much precipitation was in how arid a climate is. What's the precipitation in the dry season? What's the total annual precipitation? And what's the length of the dry season? These I thought would be relevant. And you can see only one of them turned out to have any sort of significant correlation. It was pretty strong. And it also gets stronger as we go to the more extreme climates. And this is, again, what we'd expect if selection is happening, is that this is going to be most relevant in the most extreme areas, if this is due to selection. Periodic food availability. Again, this marches toward this really strong, significant correlation as we go out to the extremes, and indicates that periodic food availability is important. and that being worm-like allows you to persist in these most seasonal areas. And lastly, the predictability just wasn't a significant result at all. The figures I just showed were for just subgenus Batrachoseps, which is the 19 or so species that's in that one subgenus. But if I include all of them, it's similar. Negative 0.73 for all the species, negative 0.8 for just that subgenus in the end. So it was similar, but you can see there's some kind of funky pattern. So I looked into this because we have that weird disjunct distribution of the Plethodontid. And with these two things, the two metrics that were significant, which was the annual seasonality and the dry season rainfall, we see there's a really big difference between the two subgenera. And let's see. And so if we think about this and what this ancestral distribution probably was, right? We saw all those disjunct distributions out of New Mexico and Idaho, and on a smaller spatial scale here, you know, that this population in Oregon we probably think is reasonably close to ancestral conditions where it's a wet, forested area. And that for Plethodontid, where they've persisted, it hasn't gotten any more seasonal, it's just gotten drier and drier and drier and drier. Whereas the subgenus Batrachoseps, where they've persisted, is where it's gotten seasonal. And so you really have this split in terms of how you think about probably their activity patterns are happening, when they're able to take in food. The subgenus Batrachoseps only takes in food in these seasonal bursts and then it's down. Whereas Plethodontid, even though it gets very, very dry, they're probably active or have a higher probability of being active throughout the year. So this is kind of an interesting split when you think about how these things develop and how these subgenera are split. So anyway, populations of worm-like forms can persist in highly seasonal areas. Generalized form gives summer relief. And the field data that was taken by this, which I feel like it's so hard to get people to give you money to go do field data now, but it was just so critical for parameterizing this approach. And when I talked about that split in the life history between the two subgenera, when we look at the osteology of these species, it looks like subgenus Batrachoseps are probably more pediomorphic. And so there's a developmental avenue to how they both develop this elongate morphology and how they and how they're persisting in these areas. Just some next things that would be cool to do. So to look at this pattern on the intraspecific level. So within a species, look at between populations. Do we see that the variation of morphology within a species tracks these same patterns in precipitation regime? And then also apply this to other species. There's a few, especially some of the skinks, since I was talking about them earlier, which are sort of similar, to apply it to the skinks. And then use some life history models to get a finer point on what's going on here. So anyway, overview, skinny winters persist in these large ranges because they can handle more seasonal habitats. And ecological differences between these species, which looked so similar, are actually best understood in the context of persistence in seasonal habitat. And that's it for today. Thank you very much. Yes, Steve.
Did you do arena tests for agonism intraspecifically? I'm just wondering if these guys really ever respond to another salamander.
I don't think they do ever. I mean, I've put a lot of intraspecific. I didn't do it formally in my trials, but I've had a lot of nigriventris together and stemata together. But I mean,
yeah, well, that makes the lack of interspecific aggression maybe not as interesting as if there was intraspecific aggression.
That's true. I agree. Yeah, it would be more interesting if they did anything. It doesn't seem. Yeah, they just don't seem to really do much of anything. I thought about this in the context compared to wetter areas. I wonder if there's too much of a cost to being mean, because if you're just not going to be able to have the resources to be really nasty, or if it's just going to be, there aren't enough hiding places, and that's the only way to be, I don't know.
Eastern plethodon live in really moist habitats, and they do show aggression.
They're highly aggressive at times, yeah. I was thinking, so Nate had a great question. I gave part of this talk at the symposium, and you were asking about using the recent climate data versus historical climate data. And so Nate had a great question. So he said, well, we're looking at recent climate data for the seasonality. Does that really relate to the evolution of the group? And I had the same thought that Nate had, which was, man, I wish I had that old climate data. I was thinking about it again, and in the context of persistence, and that we've been in a pretty consistent interglacial for what, 10, 12,000 years, something like that. So what we really probably care about is where these things are able to persist. And so, if we think that the climate was driving speciation events, I think that would matter more. But we're pretty confident that these speciation events are happening through vicariance, just because they don't go anywhere, they don't move, because there are tectonic plates or rivers forming, and that's what's splitting up the species. So I think that using the recent data, which talks about how, where they're able to persist in being arid, probably is actually pretty relevant. I was thinking about you at home, because you had a great question last time you talked about. Anyway, yeah, Angie.
It's been a while since I've thought about salamanders extensively, but these are the lungless salamanders, right? So they're doing all their gas exchange across their skin. Is the surface area by volume ratio coming into play in that regard?
So that's one of the things definitely that you're thinking about when you're looking at these things is, something that's chunkier like this versus something that's really, really skinny there's a big difference in the surface to volume ratio. And so how is that playing into, both? There's probably two things. One is how easily they can get into cracks and get down, right? So it's good to be skinny for that. But there's probably a trade-off, right? So is it better to be skinny where you can get maybe a little deeper or fit into a smaller crack, or that you have a larger surface to or lower surface to volume ratio so you don't dry out as quickly? I think those things are probably happening. And as I mentioned with the Plethodontid group, we have the two species in the south that have remained in these areas that just get drier and drier and drier. Those are our two most robust species. So those are the ones that are probably relatively close to the surface for most of the year, and they have the lowest surface to volume ratio. On the other hand, all of these things are so small and are dried so quickly that I don't know if I'm not sure how that's all playing in. But yeah, it's an interesting question. One of the things that we think they all can do, which is really neat, which is why it's good that they can get down in the soil, is they can mess with their osmolarity. So they can up their osmolarity super, super, super high, so that even if the ground isn't moist, if it just isn't totally dry, that they won't lose moisture. So then it's just a matter of finding a crack where you can not have air on you and just hunker down. So anyway.
And a second question.
Yeah.
I don't know if you showed this. Does the leg size correlate with the?
Yes.
Okay.
Yeah, in general. Now there are different ways we can go back to the. Sorry to rapid fire here. Here, that's one. Yeah, so definitely. So these things over here have actual toes. These things over here barely have any. They're just like these little paddles almost. They do have toes, but they're very small. But in the intermediate here, there is a way to be a similar score, but have one thing be a little off. So there is a little bit of variation, but in general, yeah, tiny limbs, larger limbs. There's nothing near this end that has little limbs, or near this end that has big giant limbs, for the most part. Yes, Scott.
Chris, how long do these things live?
We think they live pretty long, actually. I mean, you can keep them in captivity for a number of years pretty easily.
Yeah, has anyone tried to do a population dynamic study to find out at what times of year or life stages survivorship is lowest? Are there bottlenecks?
Yeah, actually, Virginia Majorana, that's some of the work that she did that I relied on to parameterize this, and there's pretty high juvenile mortality.
Juvenile.
Yeah.
I was wondering if there's a bottleneck during the dry season when they're.
Oh, you mean time of year? I'm sorry, I thought you meant time. Yeah, I mean, that's the big, that was her conclusion, and why, one of the things that she was focused on was that you go into that dry season and who comes out, the juveniles, a lot of them don't come out. So you have a big mortality out of that dry season.
And have they made the transmitters micro enough? I remember dealing with bees, so you put these micro transmitters on bees. Can you put micro transmitters on these and find out where they go in the dry season? If there's a bottleneck there, and sort of categorize their dry season habitat.
That's a good, I mean, I think when I first got here, I thought about stuff like that before I got more realistic about what I was trying to do. But I think that it would be really cool to know. What I worried about too was how to attach them, if you could insert it into their body cavity or not. There's not a ton of room in there, especially on the skinny species, but I don't know, I wonder about then reading them underground too. Like, would you be able to read them, and how deep you'd be able to read them? I gave up on it before those reasons, but I would really like to know. If we can figure out a way to do it, I'd be really excited. But because like the first couple of years, I'm like, we gotta figure out a way to track them, And I sort of gave up on it for this round. But man, it would be really good to know. Sarah.
Yeah, I'm curious. You have a huge expansion on vertical range of some of your species off of where they previously
Yep.
were. Do you think that's lack of sampling at high elevation in the past, or climate change, they're moving up?
I think it's like a sampling. First step is that anyway. Yeah. Good. So one of the tricky things is a lot of these lands where you want to look are privately owned or ranches, and you go out to a rancher in Kern County. Even if I'm wearing this nice shirt, they're not going to be too excited to just let me out there. Especially if you say salamander for some reason, they just don't want to hear anything else you have to say. So yeah, I think it's mostly lack of sampling. At least for this one, it's lack of sampling. And we still have, I mean, if you look at this range, that's not the best range picture. That's good enough. But if you look at, we still have gaps in here, like right here. So this is the Lake Isabella Basin, there's the Paiute Mountains. There has to be something there. We've looked a little bit, but it's really hard to get to. One of the things that happens up there is by the time the roads are dry enough, the habitat's dried out. When the habitat's wet enough, the roads are too wet, you can't get there. So there's just some very kind of real world constraints on doing it, but I think it's just lack of sampling. Nate, you had a question?
Just kind of one of my ideas I just got of tracking them down. Has anyone ever created mesocosms, like little, like habitats in a swimming pool that's 3 feet deep, and then you just break it apart when it's the dry season and figure out where they are?
Yeah, I mean, we've played around with that. I'm not. Did you mean where they're in terms of measure the microhabitat they're in, or do you mean just how deep are they going to go?
You could create an artificial environment where you could actually break up all the soil and track them. Or
Yeah.
If you found them, kind of gate off that area, say, all right, you can only bury down these 2 square meter plot.
Yeah, I mean we've done.
Remember the Cunningham paper with a 10-foot deep?
I'm not totally sure what you'd learn from having them in a mesocosm. That I don't know, what would you just have?
Give them different microhabitats in the mesocosm, figure out which one they go to, or how far they bury down. You can alter soil moisture.
Yeah. Yeah, I guess maybe you could look at differences between species and what they do something different, or they're probably going to go to the wet spot. I mean, we played with it informally. We actually have tried because it is really interesting. And there is some work on plethodon, eastern plethodon, where they did find if they gave them little tubes to go into, that you did have this breakup where one species consistently went lower than the other species. So there might be some partitioning where there's sympatric of this kind of use of these retreats. So yeah, I think there's some room to do that. Yeah, I think there's some use. So the Santa Monica story is, you go out by the LA airport and it's just sand, and then there's these pockets of trees that have oak trees, and that's where the Batrachoseps are, and there's nothing in between. And you can dig down there 10 feet and you'll never find anything in the dry season. You show up after the first rain and there's 20 of them all around. So, I don't know, it's tricky. They're really tricky to find. Yes, and Nick.
Has there been any expansion of their ranges just from development and stuff? You say there are trees near the LA airport and stuff, and that's moving
You getting transported with trees, you mean?
No, more like just more habitat and water available, I guess, because people are watering their lawns, building golf courses.
Right. In terms of how they interact with development, there's a couple interesting patterns. One is that if a city is built relatively slowly, they actually can persist. So if you go to the Mission in San Francisco, which is totally paved, they're in the Mission. They're in a backyard that's like the size of this right here. My friends have in their backyard there, they find them all the time. So these little populations persist there, but where you have just like mowing stuff down, they don't really persist. I don't know if there's been any expansion with watering, but there has been persistent. So actually, I'll give you one example. So they think there was through tree delivery. There's a species found in LA, is out in Hanford in the middle of the valley. And then they can persist in people's yards there because they're watering. So, but what's nice now with the genetics, you can catch that stuff pretty quickly, these transplanted populations. Mark?
How similar are they? I mean, I was just like, for all the stuff you showed, it doesn't seem like they move much. So for like a single guy, I guess I have 2 questions. What is, if you have any idea what a range size is for a single salamander? And if you like look at a single area on like their scale, is there any like natal dispersal, or is everybody just sort of one family like living in given pockets?
That's interesting. So I'll take the N one first, which is I don't think we have looked like I don't know if we have the tools yet to really distinguish between individuals like within like a hillside. That would be cool to know. I think we're probably getting there. I don't know, Elizabeth can probably tell us how close we are to getting there. But we haven't really done that yet. As far as how far they move, we think it's on the order of like 5 to 10 meters in their year or even in a lifetime, depending on so the studies have been, the studies were a little older now, but someone tracked all the Batrachoseps in their large yard in Santa Monica, and they basically was like the same animals were into the same stuff for like 5 years in a row. And so we just don't think they move very far. They just kind of hunker down and eat invertebrates and try not to dry out. You have a question, Elizabeth? Oh.
Is she there?
She's there.
You could ask her if she had a question.
Oh, I just did. She said no.
Oh, okay. We should probably wrap up. It's getting a little late. Thank Chris and go upstairs for some refreshments.
Yeah, we've got some beers upstairs. Thanks, everybody. So much fun.